I want to dedicate some time to talk to the importance of the actual technology used in automation. So, um, as it relates to automation and standardization within cell therapy manufacturing, what are the actual solutions and or services needed for process automation in manufacturing? And Craig, I'd like to start with you if that's fine.
That's fine. I mean, I think there's a couple different approaches that companies have made. So obviously BlueRock is fully owned by Bayer. Bayer has announced an initiative and collaboration with Cytiva so that we're actually working with Cytiva to standardize some of the common process steps, such as washing steps or expansion steps that we think are ready to be standardized and would benefit the entire industry, not just ourselves as well. And I think when you're using those type of solutions, that's a big win for all of us, and it lets us focus our detailed automation around transfection steps or around differentiation steps that are very specific to a product. We can internalize the automation and make it right for us. So I think it's taking that two-tone approach.
Okay. Narinder, how about you? What would you say are the actual solutions and or services needed?
Yeah, I think again, Craig is bringing in very important points, right? And it's about getting understanding what is important for your process and then automating that. For example, one thing we are focused on at this point very heavily is improving reliability of automated equipment. So we're working with autologous product where you are handling a patient's cell, and remanufacturing may not even be a possibility if you have to go back to the patient or their disease may be advancing fast. So in this case, you cannot rely on automation that is not highly reliable. So that's one thing. We're already facing some challenges in that and finding ways to improve reliability working with the equipment supplier. The second thing we see is the 21 CFR Part 11 compliance for the data integrity, right? In this case, as you know, the data is critical, and we need to make sure that data, can be integral throughout the process. And some of the automated equipment that are available do not have this capability. So working with those providers to get that compliance. The other thing that we are focused on is how can we use automation to to have close processing and material transfer, material handling in a more reproducible way, minimizing the risk of contamination. And we talked about at-line analytics. The last thing that I think is probably a game changer, which if we could do is, the patient cells are delicate, and these are live cells, and depending on the patient conditions, the cell quality may be different. So getting them transported to a manufacturing facility takes time. So if there's a way to cryopreserve these materials right at the source, that's going to be really important. So finding ways to automate a validated solution in a box kind of approach where it can be implemented on the hospital sites, and you could have starting patient material not only collected but cryopreserved from the get-go.
Sure. Okay. I asked Miles to post the results of the poll, and I don't think anybody on this webinar is at all shocked that scalability came in at #1 in terms of the response and what are the biggest limitations of our audience's current process. Probably what is keeping them up at night. Craig, I'd like to ask you to just provide a little bit of commentary on why you're not shocked that scalability, or maybe you are. Sorry, is the #1 response. But also, I don't think we could solve it in this hour for sure. But what are some of the things that are #1 to reaching the goal of scalability?
Sure. I mean, I think, first of all, I'm not shocked at all. And I guess I probably lost more sleep in the last 7 years than I did prior to that because of this question, the scalability. I think, look, there's a couple different challenges. One is if we are doing it lot by lot, patient by patient in the autologous world, then it means you really do have to drive down processing times so that you can get as many lots through a fixed overhead as possible. I think we talk about, it's easy to talk about that the incoming source material is variable, and that's true. But also, we're pretty nascent in how our media companies are making media. So there's a lot of variability that we're throwing into that, which causes all kinds of unpredictability in processing times that then you have to build automation around to make it scale. So I think those are the two places where I think you get immediate improvement, is making sure that you can control the raw materials so that you only have one raw material input variability. The reason why I was excited to join BlueRock is I think the way to really make it scalable is to go to allogeneic. So I think the future of pluripotent stem cells and differentiating into the right target cells gets around a big bottleneck in the world. It's not going to be ready overnight, but I think that's going to be the key to making this really scalable and making sure that we're treating patients not just in US, EU, and Japan, and maybe Australia, but all around the world. So I think those are the key areas that we're focused on.